In this video, we're going to start talking about body systems, and we're going to start with the human immune system. This is probably the most complex system. There's a ton of terminology, and there are several key processes you have to understand. So please make sure that you ask me questions if you are stuck on any of these concepts. So it's first going to be important to talk about the different types of immunity. And what I have here is a general flow chart to break down the different terms that we're going to be using. So the two main types of immunity that we discussed is innate immunity and adaptive immunity. Innate immunity is just like it's just what it sounds like. And like for example, when we talked about innate behavior, this is immunity that you are born with. This is contrast, or in other words, it doesn't rely on a prior exposure to a pathogen. This is contrasted to adaptive immunity, which is in a type of immunity that is acquired. And so this depends on prior exposure. And there are two subtypes of adaptive immunity, and that is passive and active immunity. And so depending on whether or not we're talking about a natural adaptive immunity or an artificial adaptive immunity will determine what that passive and active is. So an example of a natural passive adaptive immunity is when an embryo is developing within a female. The female, through the umbilical cord, is actually passing down antibodies. An example naturally that is active is when individuals are making their own antibodies as a result of a prior exposure to a pathogen. Artificial adaptive immunity, passive would be antibody transfer, so that's taking an antibody from several individuals and putting them into someone. For example, people can receive a shot of something called gamma globulin, which helps boost their immune system. And then an active version of artificial adaptive immunity would be immunization. And so that's pretty much taking a vaccine or a dead or a live living pathogen in a small dose and injecting that into an individual. So again, three main types of immunity: innate versus adaptive, and then you should further be able to break down adaptive into passive and active, depending on if we're talking about a natural adaptive immunity or artificial. Also, just to get more vocabulary out of the way, make sure that you're familiar with these key terms: antibody and antigen. An antibody is is a molecule that can recognize a foreign object. An antigen is anything that will evoke a immune response, and typically these antigens are going to be pathogens. So you have a couple of lines of defense before we start getting into the meat of the immune system. And the very first line of defense is anatomical barriers, and the first is your skin. Your skin serves as a protection to outside pathogens. Your mouth also serves as an anatomical barrier. We have these structures, or not structures, these molecules in our mouth called lysozymes or enzymes that break down pathogens. You have cilia in your esophagus that help push pathogens out of your throat. So when you cough, that is you expelling pathogens. And then you also have the stomach acidity. So the pH in your stomach's really low, around 4 actually, and that prevents pathogens from living. The second line of defense includes a whole host of non-specific barriers. And some examples of these are the inflammatory response, phagocytes, something called a complement system, interferons, and then natural killer cells. The inflammatory response is really interesting. Essentially, what happens is histamine is released from these cells, or released from cells in your immune system, and histamines will actually cause vasodilation, which means increasing the blood vessels. And that means more blood can flow, which means more phagocytes can go to the site of an infection. Phagocytes are cells that can undergo phagocytosis, or remember, engulfing other cells. Complement system is just a series of proteins that aid in the lysis of infected cells. Interferons do this, are very similar to the complement system, as are natural killer cells. Now we're getting into something, or now we're going to get into the meat of the adaptive immunity. And adaptive immunity essentially has three main parts to it. The first is recognition by APCs, and these are antigen presenting cells. And we're going to talk about how that works in just a minute. The second part is the activation phase, and so this is when these cells, typically B lymphocytes and T lymphocytes, are going to produce clones that are going to become effector and memory cells. Effector cells are the cells that immediately go to destroy foreign bodies, and then memory cells are the ones that stick around, that allow an individual to respond much quicker the second time they're exposed to a pathogen. And then you have your effector phase, and this can be one of two responses, either a cell-mediated or a humoral response, which is what we're going to talk about in the next couple of slides. So talking about T lymphocytes and the cell-mediated immunity. This is going to occur in several steps. So remember, T lymphocytes and B lymphocytes are responsible in adaptive immunity. So I'm going to walk you through what happens here. So an antigen-presenting cell is going to encounter and ingest a microorganism. So here's our APC, and here's an antigen, or a part of a pathogen. The antigen is going to combine with some self molecules within this antigen-presenting cells, which we call MHC, major histocompatibility complexes. And the MHC and the antigen fragment is going to be put out on the surface of that antigen-presenting cell. So essentially what happens is that cell engulfs that particle and puts it out on the cell membrane so other cells can see it. So now at this point it's pretty much saying, hey, I have a foreign object, come take care of it. These helper T cells are going to combine to the complex, and that triggers that antigen presenting cell to produce a hormone called interleukin-1. That interleukin-1 is then going to stimulate more helper T cells. It's also going to cause that helper T cell to produce something called interleukin-2, which is what's going to stimulate the T cell to produce clones of itself. This is an example of positive feedback here. Once we have several clones of the T cells, the cells are going to produce cytokines, and then these cytokines are then going to stimulate the cell-mediated immunity. So this is when we're going to go and attack infected body cells, or it's going to produce or cause the humoral immunity, which is when your body will actually create antibodies that will later on be able to recognize these antigens much quicker. So we just talked about pretty much the cell-mediated response. Now we're going to switch gears and talk about B lymphocytes and the humoral response. So it's slightly different. You have a B cell that's triggered when it encounters its matching antigen. Key term here is matching. Your immune system is very specific. The B cell is going to engulf that through phagocytosis and digest it, and then then it displays the antigen fragments using the MHC molecules, major histocompatibility complexes. This antigen MHC complex is going to help find a matching T cell. So the T cell is then going to come and bind to that B cell. The cytokines secreted by the T cell are going to help the B cell multiply and mature into antibody-producing plasma cells. So this is important. B cells produce antibodies. And then those antibodies are going to be released into the blood, and they're going to move around the body throughout the blood, and they are going to go and target those antigens. So all of these antibodies are made to target that antigen or that foreign object that was introduced into the body. What happens then is, because those antibodies bind to the antigen, we activate the complement system, or the complement cascade, which is that series of proteins which tell those cells that they need to lyse, or it goes and destroys the pathogen. So slightly different outcome here when we're talking about the humoral response. There are 4 general characteristics of the immune system. It's very specific. It's very diverse. You have several different types of cells. We have memory, so what this allows us to do is respond quicker each time, because remember, in that activation phase, we're producing effector cells, which immediately go and target that pathogen, and then we also have memory cells, which stick around and allow the body to respond quicker next. And then it's also very important to describe the fact that the human immune system has the ability to determine self versus non-self. So essentially, your body can immediately recognize when something is there that should not be there. It's also related to the fact that your body has several mechanisms in place that prevent your body from targeting your own body cells, and so that's a term we use to describe that is called self-tolerance. And then towards the end here, I just want to point out a graph. You should be able to read this graph. You'll see something very similar on your test, I promise. But what you can see is along your x-axis is time, along your y is antibody concentration. So remember, in your very first, or the primary immune response, you don't have memory cells. The cell-mediated and the humoral response have not occurred. And so it's going to take a pretty long time for your body to fight that pathogen. So after the initial exposure, long time, and the concentration of antibodies is not very high. However, because you have all of those memory cells in place, the second exposure to an antigen, what do you notice happen? At a much quicker, so in a faster response happens, and the concentration is much larger. And so this is a cyclic graph that you'll see comparing primary to secondary immune response.